I'm Bernard. And I'm Sophia. And today we're going to talk about enzymes. Yeah! Hi, biologist. Okay, so today we're going to talk about one of my favorite molecules ever, the enzyme. Now, what makes enzymes so special anyway? Well, enzymes are proteins. Remember all our organic compounds? Well, enzymes fall under the category of proteins. So that means they're made of a long chain of amino acids. Amino acids, building blocks of protein. Don't forget it. So, enzymes are really cool because they are the catalysts of biology. They make things happen. They get things going. They get stuff started. So, an enzyme is going to work on a biological reaction to make it go faster. Now, an enzyme wouldn't get anything done that something normally wouldn't have a reaction for, but it does make the reaction happen a little bit easier. So this is just an image to get the idea of an enzyme better in your heads. So you can see here, this is our enzyme, and if you look at a small bit, you can see it's made up of a long chain of amino acids. This part right here is what we call the active site, or active sites. This is where the enzyme is going to bind to another compound and get the reaction going. Now here are the basics of how an enzyme works. We start with the enzyme and the reactant, the thing the enzyme is going to work with. We can also call this the substrate. Let's write it down here, the substrate. Now the enzyme is going to fit a specific substrate at its active site. And you can see when they're together, we call that the enzyme reactant complex or the enzyme substrate complex. Now, when the enzyme is finished, the enzyme looks exactly like it did before, but the products now look different. So, for example, an enzyme is going to take something like Bernard here, a substrate, and make him do something that he would normally do already, but the enzyme's going to get him going. So, pretend I'm Bernard's boss. Say, "Hi, Bernard. Hello. Bernard, you need to do the rest of your work." Okay. Enzymes can also work on two different substrates. So, Susie here is my friend, and Bernard, or Sophia, I don't remember your name. I'm Sophia. So, Bernard here is my other friend, and if Sophia and Bernard met, they'd probably get along. Oh, hi, hello. But they are two people in a very large city, so they might take a while to actually meet, run into each other, and make friends. So I, the enzyme, am going to introduce them. I get this reaction started, they're going to be happy friends, and live happily ever after. Now, enzymes are often called molecular scissors. So sometimes we have really long molecules enter the body that we need to work with, but they're so long and they're going to take so long to break apart on their own that we need an enzyme to help us with the process to get this cut into smaller molecules. Now the scissors here are the enzyme working on the long molecule. You can see it's broken down into smaller parts. The enzyme at the end remains unchanged. Nothing happens to the enzyme and it can keep cutting away. Now remember, enzymes are highly specific, so one enzyme can't work for a different type of molecule than it normally works for. Enzymes are also very constant. After the reaction, the enzyme will stay unchanged. The substrate will go away, forming the product, but the enzyme is ready to go again and make something new. Go! Enzymes are a lot like professional athletes, and environmental factors can affect them. So there are a lot of factors that can affect the activity of an enzyme. Now, temperature is one of them. If the enzyme gets too cold, we know as temperatures decrease, molecular activity is going to go down too. So the enzymes will be moving slowly around the cell and the reaction won't be as fast. If the enzyme gets too hot, remember the enzyme is a protein and the proteins are made from amino acids. So those amino acids are going to start to unfold at certain temperatures and the enzyme will lose its shape. So if it loses its shape right here, it won't be able to react with the substrate, and again, its activity will go down. So temperature is one of the factors that can affect the enzyme. Now, pH is another factor that can affect enzyme activity. We've already talked about how it's important to maintain a stable pH in the body, but different enzymes work better at different pHs. There's some enzymes that live in the stomach and work better at an acidic pH. There's other enzymes that prefer a basic pH. But if something goes beyond or below the pH that the enzyme is used to, or its optimum pH, the activity won't be as high. So if you see on this graph, as the activity increases, we'll have a preferred pH for that enzyme. The increasing pH, so it's getting more basic down here, more acidic down here, and right here, this is the range where the enzyme is going to function best. We have lots of different enzymes in our bodies that make things happen. So, some enzymes are digestive, some enzymes are regulatory, other enzymes do other things. But to recognize them, enzymes are almost always have a special 3-letter code in their name, and that code is ACE. A-S-E. If you see a word with ACE at the end, it's probably an enzyme. There's a few exceptions to the rule, but most likely if a molecule ends in A-S-E, like amylase or maltase or lactase, that's going to be an enzyme. So enzymes are useful in the body not only to help us with normal reactions, but also to get rid of toxins. Now this is the enzyme ADH. It helps us when ethanol, the ingredient in alcohol, enters the body.
A colorless inflammable fluid with a characteristic odor and a pungent taste. Chemical formula C2H5. You know it as alcohol.
Now our enzymes aren't perfect molecules. There are about enough ADH enzyme molecules in your liver to take care of 1 drink's worth of alcohol. However, if you drink more and more, there aren't enough enzymes to work with all the ethanol molecules and metabolize all the alcohol. The special enzymes can't deal with it all, and because ADH enzymes are very specific to alcohol, all of the other enzymes in our bodies can't help out. So the alcohol that is not being taken care of by our ADH enzymes leaves the liver and ends up in the bloodstream, leading to a higher blood alcohol concentration, or BAC. As the alcohol flows through the body into the bloodstream, it continues to work as an active drug, producing all of the classical physical side effects of intoxication. Okay, I hope you've learned a lot about enzymes, but before we go, I want you guys to look up on the internet and try to find 5 enzymes that are used in digestion. 5 digestive enzymes in the body. We'll see you next time.